Hello, everyone. My name is Camila Esguerra, and I head the Chemical Neuroscience Group at the Center for Molecular Medicine, Norway, at the University of Oslo. It's a pleasure to be here today and to share some of the research that my team and I have been carrying out in the last several years. My aim is to demonstrate the power of using the zebrafish to model neurodevelopmental syndromes and brain cancer. I will present two use cases to show how we have utilized some of the unique advantages of the zebrafish model to model CNS disorders and how we use these models to elucidate early disease mechanisms and also use some of them for preclinical drug discovery. So, what is our research about? My team and I are focused on understanding neurological disorders, in particular epilepsy, schizophrenia, and autism. We generate new animal models for these disorders using genetic engineering to introduce specific mutations or chemical exposure during neural development. And more recently, we use tumor cell xenografting to study cell-cell interactions to see if we can trigger epileptogenesis by altering the host tumor microenvironment. In particular, we focus on disorders known to be refractory to anti-seizure medications and use these models to understand the underlying disease mechanisms. In uncovering new disease mechanisms, we hope to identify new avenues for therapeutic intervention. Some of these models are then selected for carrying out large-scale drug screens, and once drug candidates are identified, we use our animal models to understand how the drugs work, in parallel testing drugs for potential toxicities or adverse side effects, as the zebrafish is also an excellent model to carry out such analyses. My team and I have built up a research platform over the past years in Oslo using genetic engineering and pharmacological methods. We generate disease models in zebrafish and analyze the phenotype using a variety of assays, including automated behavioral assays, EEG and AI-assisted analyses of seizure patterns or brain recordings, also calcium activity now using 2-photon laser microscopy. And we also perform imaging, pharmacological profiling, and omics analysis and histology to try to understand what's going on as a result of these mutations that we introduce into the fish that are modeled after actual patient mutations in order to validate our model. Ultimately, we will then select some of these then for large-scale drug screens. So why zebrafish? Zebrafish pose many advantages as a model organism. It is a vertebrate with a fully sequenced annotated genome, and with the exception of lungs and a uterus, they have strong genetic, physiological, and pharmacological similarity to humans. They are also highly fecund, which means they're very fertile and great for genetic studies or large-scale drug screens or phenotypic screens. And basically, a single mating pair can produce up to 300 embryos in a single spawning session. They're also very small in size. So this is an adult, 2 to 3 centimeters in length, and a one-cell stage embryo, newly fertilized, and it's only about 0.6 to 0.7 millimeters in diameter. And because of their small size, we can then assay them in 96-well format. They also develop very rapidly ex utero within 24 hours, and you basically have a body plan with eyes and the brain, and even insulin being produced by the beta cells of the forming pancreas. And in about 30 hours, the heart starts pumping and the blood starts circulating. They also develop rapidly ex utero, which means we never have to sacrifice the mother for our studies. And probably the biggest advantages of the zebrafish is its optical transparency up to 2 weeks after fertilization, which means that we can perform non-invasive imaging in real time. And for compound screens, we only need microgram amounts or sub-milligram amounts of compounds, and they are readily absorbed in the medium in which we bathe the embryos and larvae. And for CNS studies, they are diurnal like humans. So the zebrafish is great for studying CNS disorders because its overall brain organization is very similar to humans. So they have an olfactory bulb, hypothalamus, lateral pallium, which is the equivalent of hippocampus, and a cerebellum. There are, of course, some differences, such as a smaller cerebral hemisphere, and there are some differences in the forebrain layout due to the way the brain develops. And also some differences in the structure and function of the optic tectum. However, the main neurotransmitter systems are present very early on, from cholinergic, dopaminergic, noradrenergic, GABAergic, and etc. The blood-brain barrier is functional by 10 days post-fertilization. And as I already mentioned, they have a sleep-wake cycle similar to humans, which means that they're awake during the day and asleep at night. And many neurological disorders have already been modeled, including epilepsy, Parkinson's disease, Alzheimer's, ALS, ADHD, ataxia, autism, and even schizophrenia. So for those of you who are not so familiar with the zebrafish brain, this slide beautifully illustrates the advantage of its small size combined with optical transparency. The upper left panels over here in green and red basically demonstrate that the use of fluorescent transgenic reporters can visualize different neuronal networks and pretty much any part of the zebrafish brain, and you can image the whole brain. The video I'm about to show you is courtesy of the Aaron's Lab at Genelia Farms, and here you can see that you could also visualize brain activity in real time in the whole brain. Here you see basically the different activity patterns being recorded. So a few facts. The 5-day-old larval zebrafish has a brain that is only 800 microns long and half as wide and 300 micrometers thick. At this point of development, it has about 100,000 neurons, and many of the neurotransmitter systems are already mature and active. The bottom panel here shows the human brain in beige, the mouse brain in orange, and the adult zebrafish brain in gray. So as you can see, in terms of scale, how tiny the zebrafish brain is relative to mouse. So here, we can also record different types of behaviors. And what I'm about to show here in these videos is basically convulsive-like behavior when the larvae are seizing. These are 7-day-olds in single wells of a 96-well plate. And what you see in the left is basically wild-type sibling control in normal embryo medium. And it's still feeding off of its yolk sac. So it's trying at this point to conserve energy and isn't very active. So see here. But if you zoom in, the heart is beating nicely and it will move every now and then. In contrast, if you bathe same wild-type 7-day-old larva in pentylene tetrazole, which is a proconvulsant compound and essentially acts as an antagonist to the GABA A receptor, you'll see that it goes through different stages where it becomes very hyperactive. And eventually that's what we call stage 1 seizures. And eventually it will then demonstrate burst activity in this whirlpool-like movement, which I believe is coming up soon. And see some twitches as well in the tail and fluttering of the fins. There we go. That's stage 2, whirlpool-like behavior, as we call it in my lab. And then eventually it'll reach stage 3, where it is no longer able to remain upright. So it loses its posture. And this is reversible. So you can wash it out and put it in normal medium, And after a few minutes, the larva is perfectly fine. So for epilepsy, the golden standard is still to be able to record from the brains. And in order to, because a lot of things can cause hyperactivity. And of course, if we want to confirm that the larvae are actually seizing, then we perform local field potential recordings where we place the larva in a small microscopic slide and cover it with a very thin layer of soft agar in order to hold it in place. And of course, in its embryo medium bath so it doesn't dry out. And then we carefully place a fine microelectrode into a select region of the brain, and then we can record the local field potential around where we place the electrode. And what you see here is the optic tectum because it's very easy to locate and the neuronal layer is near the surface and therefore easy to access. And these are typical recordings in normal vehicle control, so negative control, that's what establishes baseline. And then in the presence of pentylene tetrazole, and then in combination with other drugs. So some drugs, such as phenytoin, will actually exacerbate PTZ-induced acute seizures, whereas diazepam and other drugs, tiagabine, will attenuate the seizures, as you see here in the trace recordings. So just prior to the COVID-19 pandemic, we published an article in the journal Epilepsia describing new insights into the very early mechanisms underlying an epilepsy syndrome, in this case, Dravet syndrome. And we tried to find the very early defects in the developing brain as a result of mutation. And just briefly, I want to mention what Dravet syndrome is. It is a very severe genetic epileptic encephalopathy with early onset between 6 to 18 months of life and often triggered by a fever, but not always. The primary gene mutated in about 80% of cases is SCN1A, which encodes the alpha subunit of the voltage-gated sodium channel 1. It is categorized as a pharmacoresistant or refractory epilepsy, and if seizures remain uncontrolled in some patients, they can have up to 100 seizures a day. Some of the comorbid features are autism, intellectual disability, social behavioral problems, and sudden death. Fenfluramine is one of the drugs that has been approved now for the treatment of Dravet patients, but was still pending FDA and EMA approval at the time of this study. And it acts as basically a serotonin receptor agonist. And I'll get back to that in a minute. So we created the SCN1A mutant using CRISPR mutagenesis. We introduced a missense mutation that perturbs sodium transport in all channel isoforms. The homozygous mutants displayed about 7 to 8 seizures per 20-minute recording, and the heterozygotes 3 to 4, so half as many seizures. Spectral analysis of the EEG recordings or LFP recordings confirmed increased excitability overall in the brain. The larvae displayed convulsive-like behavior, but ultimately, as they developed, their locomotion became more and more impaired. So they were not able to have a normal touch response and lost their posture. And when we tested a whole bunch of anti-seizure medications, the larvae actually responded in the same way as patients. So it had a very similar pharmacological profile. So this is the actual brain recording from 4-day-old SCN1A homozygous zebrafish larva, just to give you a sense of what we see when we're performing the recording. So we videotape the larvae to look at their behavior while we are recording the seizures. So we can record both simultaneously. One of the findings in our study was a significant reduction in dendritic arbors, so the branching of GABAergic neurons. And GABAergic neurons play a very important role in epilepsy because you need that inhibition and excitatory balance. So basically, they have to be equally balanced. And often, when there's hyperexcitability, this could lead to synchronous firing of neurons and ultimately seizures. And if you impair GABAergic or inhibitory neuronal signaling, either by a reduction or death of GABAergic neurons, or in this case, the reduction of the branching of the neurons and the neuronal network, then you reduce the inhibitory tone, and therefore the default is hyperexcitability. So when we crossed our mutant with a reporter line for GABAergic neurons, shown here in red, in the wild type, you can see a lot of branching already at several days. So basically, even before seizure onset, as early as 2-3 days post-fertilization. And then when we looked more closely, we could see large patches of dendritic arbors were simply missing in the mutant. And we quantified this and counted that by the time of seizure onset, which in this mutant is 4 days post-fertilization, 40% of the dendritic arbors in the GABAergic neurons were gone. So interestingly, this reduction of dendritic arbors was already present prior to seizure onset, which made us, was sort of an indicator that the structural or morphological difference in the early brain could be a possible mechanism which caused the seizures to begin with. And then, since serotonin signaling is important for neuronal branching, we asked ourselves whether if we were to treat the mutant larvae in fenfluramine chronically, so over several days of development, prior to and during the course of epileptogenesis, when seizures begin and recur, maybe we could also rescue the neuronal phenotype. And indeed, this was the case. So as you see in the left panel, wild type branching, SCN1A, untreated larvae, and then those treated with fenfluramine were basically reverted back to normal morphology and total number of dendritic arbors. So now on to use case number 2. This is in collaboration with Sebastian Watzap's group, who is at the EPFL in Switzerland, and Carl Koschmann, who is at the University of Michigan in the United States. So we have been trying to model a very aggressive pediatric brain cancer that forms mainly in the medulla or the brainstem. And we do this by transplanting patient tumor cells. And first of all, I just want to go through the background and rationale as to why we were interested in this. So tumors can trigger seizures. And so from our interest, we were very interested in what all the molecular mechanisms are that cause seizures. And so we thought this might be a nice way, by basically engrafting tumor cells into the developing brain, whether we might be able to trigger seizures. And seizures have also now been shown to modulate tumor growth. So basically studying the host tumor microenvironment and the interaction of the brain and the tumors in the context of a seizing fish is very, very interesting for us in terms of understanding what's going on. So the optical transparency of the larva, again, and into early juvenile stages, allows them for us to image and follow the tumor growth over a long period of time, and also in combination with drug treatments. And of course, the genetic tractability of the Zebrafish model also allows for disease-modifying pathway analysis, and the amenability of zebrafish larvae for drug screening especially drug combinations, which is very intensive in rodents, was basically another big advantage for using the zebrafish. So over the years, there have been multiple reports by various research labs showing that one can indeed engraft different types of tumor cells into the developing zebrafish and into different tissues. So in the hindbrain, melanoma has been engrafted successfully, as well as colorectal cell cancers into the circulation. So this is a big vessel that feeds into the heart, into the rest of the circulation. Breast tumor cells have been engrafted, and into the caudal vein here in the posterior part of the larva. There have been basically patient-derived leukemia cells transplanted as well. And in our case, we are transplanting different types of brain tumors, such as glioblastoma and these pediatric brain cancers. So this is an image from our lab, whereas we use basically injection or engraftment using fine needles carrying tumor cells that are here fluorescently labeled into a 2-day-old larva. And this is an image taken 1 hour after engraftment. In this case, we had GFP labeled patient tumor cells, and we engrafted it into the hindbrain. And the upper panels show the cluster of tumor cells in green 1 day after injection or 1 day after engraftment, and also in the bottom 4 days. And after, at around 4 days, you can see then that the cells are starting to spread within the developing brain. One can also visualize these tumor cells and their behavior and migratory pattern using confocal imaging. And this is a 5 days post-injection. And as you can see, whereas the cells were engrafted in the upper part of the ventricle, they have now spread rostrally and caudally, and also down into other parts of the developing brain. Then when we determined that the tumor cells we were interested in actually thrive and can spread in the developing zebrafish, we then tested with candidate drugs that are about to enter clinical trials for the treatment of these pediatric tumors. And what you see in the top is basically the controls. So untreated, E3 is simply the short term for our embryo medium. And this is normal light microscopy in the left, and then under fluorescence microscopy, and then combined, where you can then see the spreading tumor cells in the brain over time because they have not been treated with any drug. Whereas in the bottom panel, 5 micromolar drug treatment, you can already see that the clusters of tumor cells is much smaller, and they essentially don't appear to spread. Nevertheless, they don't die. So clearly, this drug keeps them in check, i.e. from proliferating, possibly metastasizing. However, the tumor cells still remain. And so the idea is to refine the drug treatment, so letting the tumors grow, for example, to a larger size and then treating after to simulate more of the patient scenario, but also then taking a matrix of candidate drugs and doing a lot of combinatorial drug treatments to see which combinations will ultimately kill the tumors. And so this is very exciting, and this is ongoing now in my laboratory. And we are then comparing notes with our collaborators who are computational biologists and also rodent cancer modelers, and then basically hope to combine efforts and use the advantage of each and every model in synergy. And with this, I'd like to thank you for your attention and hope that I've been able to show you some of the exciting things that we can do using the zebrafish model. Goodbye.